Okay.
This is part of the formality, I suppose, of introducing your student. And so it's my immense pleasure to introduce Emma Hudgins, who's performing her exit seminar for her PhD. Emma has been a true pleasure to have in the lab, in addition to being outstanding academically and in terms of scientific research. She has definitely made the lab a better place, and it's been excellent having her there. It would be quite sad to see her actually finally finish up, even though I'm very happy for her as well. All right, on that note, I'm looking forward to seeing her presentation.
All right. Thanks so much, Brian. That's really sweet. And it's been a joy to have been in the lab, and I'll be super sad to go because it's so important to have a good lab environment and one that fosters the growth that I've been able to have during this time and just the flexibility. And I really appreciate just having been able to be a part of this lab during my PhD. So hi, everyone. Thank you so much for joining me today virtually for my PhD seminar, which is typically a social event that's followed by a reception. So to try to simulate that, you're free to stay on the call afterward and raise a glass if you'd like, but I understand it's the middle of the workday. And even though I'll miss the social aspects of this seminar, I'm super grateful to have people from all over the world tuning in today who wouldn't have otherwise been able to make it, including my sister who's on the line from Argentina, the rest of my family in New Brunswick and BC, the rest of my committee, and all my friends from all over, and some of my collaborators too. So it's really amazing. And I'm going to be talking to you today about a project I started back in the final year of my undergrad in 2014. So I've been working with the same data set since then. And the two of us have been through a lot together. And throughout the process of working with Brian Leon, my supervisor, and the lab, I went from thinking I wasn't the kind of person who would be good at programming to someone with a good amount of confidence in my quantitative and my computational skills. And hopefully the end of my PhD goes smoothly so I can apply them finally to a different data set. My PhD thesis is entitled Predicting Forest Pests, Generalized Models for Invasive Species Establishment, Spread, and Economic Impacts. And I've been working under Brian's supervision as well as with the advisory support of my two wonderful committee members who are tuning in today, Jonathan Davis and Patrick James. So I've focused my work on invasive species, which are a stressor characteristic of the current geologic period known as the Anthropocene. And it gets its name because of the role of humans in fundamentally altering biogeochemical processes on Earth. Invasive species are species transported outside of the areas in which they've evolved, and I'll be focusing on those moved by humans. And species invasions are just one dimension of a complex set of accelerating ecosystem stressors characteristic of the Anthropocene, which has been called the Great Acceleration. And so you can see, just like human population is exploding, we're seeing accelerations in atmospheric CO2 levels, species going extinct, and major climatic events like floods. And at the same time, invasions are accelerating and show no signs of slowing down. And since all of these anthropogenic stressors interact, we need to consider them together when we're trying to forecast our future. For instance, species can invade through accidental transport in the global shipping network, and shipping traffic is expected to change into the future and accelerate to meet the needs of a developing world. At the same time, the climate is projected to change. And if species are being accidentally transported through this network and the climate is changing in ways that make them easier to establish, then we can see accelerating invasions for this reason. And so different members in my lab have been focusing on these questions, and they're especially salient now in the coronavirus era, because you can think of an analogous system where air traffic patterns are accelerating with the developing world, and that's changing the spread of pathogens. Notwithstanding potential future accelerations, we already know that invasive species cause hundreds of billions of dollars of damages annually, and insects alone make up tens of billions of dollars of these damages. And so this makes the need for effective policy to manage these species where they have invaded and limit future invasions dire. So we've established that action must be taken quickly, but to make effective decisions, we need an accurate picture both of current invasions and how they're going to progress into the future and cause associated damages. And the challenges with making these predictions at large scale is that the data are often very poor because invasions tend to be studied on a really case-by-case basis. And so my thesis chapters build off one another to tackle this issue of data scarcity and increase the resolution of my models. And furthermore, my thesis aims to leverage the role of humans in dominating these ecosystems in order to make better predictions. And to do this, I predicted that human influence means that at broad scales, invasions are behaving in less traditionally ecological ways. And I call this anthropogenic replacement because this means human influence is replacing conventional ecological processes. In other words, heavily human-altered ecosystems are being governed more by patterns of human behavior than intrinsic ecological rules that we typically think of like species interactions. And the focus on anthropogenic replacement to guide my models has allowed me to balance their management utility with a search for general macroecological rules. And these are large-scale principles that govern ecosystems in this new era of human dominance. So since my honors project during my undergrad in Brian's lab, I've been studying American invasive forest pests. And this is a specific group of invasive species that harm forest products and cause both ecological and environmental and economic damages. And here I'm showing their distribution as of 2009 from a paper that my collaborator Andrew Leopold wrote in 2013, where darker green indicates a greater number of pests in each county. And you can see they're concentrated in the Northeast and along the West Coast. And this coincides with areas of high human activity. We can group these forest pests into 4 feeding guilds based on the way that they kill their host tree species. So we have wood-boring species that bore holes into the wood of the tree, defoliators feeding on the leaves, sap feeders feeding on the sap, and then pathogenic species comprising fungi and some other disease complexes. And some typical examples of each of these are emerald ash borer, which is a major problem here in Montreal. It kills a lot of our street trees. Gypsy moth, major defoliator, also Molliadelgid, and chestnut blight fungus, which has been responsible for the death of most American chestnut trees in the US. And you can see that these species span a large portion of the evolutionary tree because we have fungi and insects. So I've been studying the secondary invasion process of invasive species, and this is what happens after they're introduced to a new location. Typically, we think about the invasion process of being broken into 3 stages. So pest is introduced unintentionally or intentionally to a new location outside of where it's evolved. It can then either establish or fail to establish, and most species fail. And establishment is defined as having a reproducing population in a new site. And then it can spread to subsequent sites. So the secondary invasion process is this subsequent spread, which looks a lot like the overall invasion process, but happening at a local scale. So each place where it spreads to next, it arrives and either establishes a reproducing population or doesn't, and then can spread to new sites. And locations can experience impacts of invasive species at any point along this process, both ecologically and economically. So the primary goal of this thesis was to identify large-scale generalities across species invasions in this secondary invasion process for use both in predicting the outcomes of establishment and spread, as well as forecasting economic impacts. So in chapter 1, I built a model for the spread of all these species across the US. In chapter 2, I compared this general model to more complex species-specific models. In chapter 3, I built a model for forest pest establishment in order to determine the relationship that host biodiversity has on pest success. And in chapter 4, I created an updated economic estimate for damages due to these forest pests to urban trees. The more complex spread models from chapter 2 are based on those from chapter 1, and the spread models from chapters 1 and 2 are used in subsequent economic impact models to fill in missing data and make higher resolution predictions. And they're also used, the spread model predictors are also used to model establishment. In my first chapter, I focused on the spread phase and built a general model for how all these species are going to spread across the US. And again, this was done in conjunction with Andrew Leopold, or Sandy, who is in the audience today and has been such a reservoir of pest expertise. He has so many years of experience with this system, and I'm so grateful for all of his collaboration with me, even though we have never actually met in person. This is actually only our 2nd time video conferencing. So hi, Sandy, and thank you for everything. So we fit our spread model to 64 pest species, and our spread model examined general predictors grouped into 3 categories. So first, habitat characteristics that might accelerate dispersal into and out of sites, which we call habitat invasibility traits. Secondly, human factors such as population density and income that might predict spread, because humans are the main vector of the transport of these species, and humans with more money tend to move around more, and they tend to move around high population density urban centers. And then as a third component, pest life history characteristics could be important for predicting spread rates. For instance, species that can fly might be able to spread faster than those who cannot. And we tested two hypotheses. First, we hypothesized that the overall pattern of pest spread is generalizable. And secondly, we hypothesized that these three types of predictors could be meaningfully incorporated into this general model and lead to improved predictions. So we fit our model in a spatial simulation framework. So we just had a big grid that represented the entire US. And in each grid cell, we had a potential population of pests. And each time step, we allowed them to disperse amongst the cells according to this equation here, where the probability of dispersal between the sites decreased with the distance between the sites and could also depend on additional factors Z. And Z was where we brought in our three types of predictor variables. So we had species-specific predictors at S, and then our habitat and human-related variables, which we included both as factors increasing dispersal or decreasing dispersal into sites and influencing dispersal out of sites, ZO. In the simulations, we started just looking at this from a more visual perspective. We started the pests at the beginning of the simulation in the center of their host range at the date where they were first detected in the US, and then allowed them to spread across time to create some sort of predicted distribution, which we then compared to the real distribution and saw how well we were able to recapture that spread pattern, both in terms of how far pests are spreading and where they were spreading to. Here I'm showing the results of that best fitting model. And whenever I'm representing an R-squared, it's a proportion of the variation explained. So our predictions are on the x-axis here, our observations are on the y-axis, and if we perfectly recaptured our observations, we would get an R-squared of 1. So we get over 3/4 of the variation explained here. And in our best model, forested land area and human population density were included as positive terms, increasing dispersal into sites, and human population density and tree density were negative terms, decreasing dispersal out of sites. So pests were attracted to areas with a lot of people and trees, and once they got there, they stayed there. They dispersed less out of those sites. And we were not only able to capture how far pests spread, but we had 74% locational accuracy, so actually getting the sites precisely correct. And here I'm showing an animation of our predicted spread patterns over time. You can see our data set spans from the 1800s to today, but most of the spread is happening at the very end of the simulation where it increases a lot. So based on the results of the GDK, we first developed the idea of anthropogenic replacement, where natural ecological processes are essentially being overridden by anthropogenic ones, and predictable generalities that operate across entire groups of species arise as a consequence of this and make the system much more easy to predict. But it was not clear just how much predictive power we lost by doing this compared to actually leveraging that pest-specific expertise in building customized models for each pest species. So that motivated our chapter 2, which we then worked in collaboration with Andrew Leupold with. And in this chapter, we compared our general model to customized models fit to single species where we had precise time series of their detections across time, and compared also a third type of model that was sort of an intermediate between the two, to leverage some of the advances of the general model, but also included species-specific corrections. So starting out with these customized single species models, we were able to fit these only to 3 species where we had these single species time series data. And these were beech bark disease, gypsy moth, and hemlock woolly adelgid. And they're some of the most damaging pests. And so these are the only species for which such a model is possible. And so models built in this way can't really be extrapolated to the rest of the community. And with these models, we use the same general framework for modeling as the GDK, but instead of fitting on across species, we fit to the single species time series. And we tested some additional flexibility here by trying out different dispersal equations and starting the pest not just from the center of the host range, but different starting points, and also looked for additional predictors, things like campground density for gypsy moth, which are known to be important, and trying those out as well. And then we fit our intermediate model type, which we're calling semi-generalized dispersal kernel models, or SDKs. And these included 3 additional layers of complexity, but they didn't rely on that time series. So they could be fit for all species in our community. And firstly, we tested including a species-specific spread intercept. And the logic of this is that, so we know from the GDK that we get the spread of some species better than others. And for certain species, we're over-predicting their spread rates, and some we're under-predicting these spread rates. And if these were general patterns that we thought were going to be continuing into the future, then we could use them predictively. So if we always know that we get the spread a little bit too fast, we can offset it and bring it down to the acceptable level and use that for forecasting. We were actually able to rely on some of very basic concepts from diffusion, from physics to do this, where we were able to refit the intercept based on the diffusion coefficient from physics. And this diffusion actually underlies some of the most fundamental ecological models for spread. So it was really a nice way of showing how very old first principles theory could be updated for conventional purposes. And in addition to the species-specific intercept, we also tried out those different starting point corrections. And then as a third layer, we tried different climatic corrections for species where we thought they might be climatically limited. So hemlock woolly adelgid is known to be limited by low winter temperatures. So we tried limiting its range to areas above a certain minimum winter temperature. And in all cases, I fit to the year 2000 and evaluated my models in their ability to predict subsequent spread in 2005. So here I'm going to start showing the results of this. In all these cases, the host range is shown in this beige color. So this is the potential area the pest could spread to. The blue is where it has actually spread at 2005, and then the red is where we predicted it spreads in 2005. So in this case with gypsy moth, we get the pattern quite wrong with the customized model, but we get the extent of spread decently well. So it has spread about this far, but not to those places. Comparing that to the GDK in the next slide, we actually do a little bit better in terms of the pattern, but we actually overpredict spread by quite a bit. So that's why we're seeing a decline in our R-squared metric, which again would be a value of 1 if we did perfectly well. So we do see some improvement of the customized model, but strange spatial patterning. Looking again across, now across species, gypsy moth predictions are in the left column, hemlock woolly adelgid in the center, and beech bark disease on the right. Across the board, the customized models do tend to perform better. They're 17% better on average in terms of spatial variation. But the GDK, considering it has no species-specific terms in it, performs quite well, especially in terms of the spatial structure. And it has 57% variation explained on average. When I compare this instead to the SDK, we see even further improvement above the customized model. And this is really interesting because the SDK does not require the time series, but the customized models do. So we actually see 17% improvement with the SDK across species, with a mean of 91% spatial variation explained. And this is great news because that means that we can use this model predictively for all species, not just the 3 with time series. And so then we do that and we create forecasts for the entire pest community up to 2030. And when we look at this, you see the spatial pattern of pest richness, so the number of pests in each site is about the same, but it's getting much more concentrated in high numbers in the Northeast. Looking at this in a slightly different way, I can subtract these predictions from these ones, and then we can just look at new establishments between 2005 and 2030. So these are new locations where pests are present. And I can group these basically into 2 categories, and these are new establishments driven by terms in the spread model, either terms related to forests. So region K is a big forest in the US, whereas some new hotspots are driven more by human population growth. And so this is the Chicago area, which is going to continue to grow, and we're going to see a lot more infestations there as well. So this is, to our knowledge, the first comparison of general and species-specific models for spread in terms of how well they can forecast into the future. And so this is a really good demonstration that we need to be testing that out much more often. But also our strong predictive results suggest that we don't always need these time series if we want to make fairly decent predictions with limited data. I know that the forecasting window is only 5 years in the future, but if we're just trying to plan for the next 5 years of management, we can do a very good job without that single species time series. And this also means that we can forecast species outside of our data set, predict new species spread patterns. And the SDK dispersal forecasts were then used in Chapter 4 in my economic impact assessments, whereas in Chapter 3, rather than just using that species-specific intercept that I used in this chapter, I wanted to try to figure out why species are differing in their underlying spread rates and try to disentangle those factors and see if any of them are related to the quality of the hosts across their invaded range. So that brings us to my third chapter. In this chapter, I shifted from the spread to the establishment phase of invasions, and I've been collaborating with my committee member, Jonathan Davies, on this work, and he's been a great source of expertise in understanding the complexities of phylogenetic analyses for this chapter. And so here I'm trying to figure out if host biodiversity plays a role in pest persistence in this system. So from chapters 1 and 2, in the comparison of general and species-specific spread models, we know that different species spread at different rates. Some spread faster, some spread slower. And rather than just summarizing this in an offset or an intercept that changes for each species, I wanted to see if we could tease apart some of the mechanisms for this. And similar to models of spread, biologists can spend years creating really precise establishment models for a single species based on really species-specific expertise. But again, in the general motivation of creating general models that allow us to respond quickly to invasions and also new species with limited data, I wanted to see if there are any general large-scale predictors of this. And specifically, I wanted to look at the quality of the host range of these pest species, and in terms of the types of species, host species present, and how many there are, and see if there are any of those general spatial predictors of pest success. So hosts, and in this chapter, whenever I say hosts, I mean trees, and then the pests infest the trees, so the pests are infesting those tree hosts. But it's going to be a little bit confusing because I'm also talking about phylogenetic trees. And the phylogenetic tree is what's represented here. So hosts are known to be differentially susceptible to pests based on their evolutionary history, and we describe evolutionary relationships frequently with phylogenetic trees, where the length of the branches between the species describes how long ago they shared a common ancestor. So species D and E are really closely related, whereas species A and F are quite distantly related evolutionarily. And more specifically, hosts that have no experience with an invasive pest, but that are closely related to hosts within the native range of invasive species, are more likely to be the most susceptible. And this is because the probability that a plant species is susceptible to a particular pest declines with evolutionary divergence between the pests. So in this figure, it's looking at something called pathogen spillover, which is basically pathogens of these other species then going on to infect species C here. And pathogen spillover becomes less and less likely as you get further away evolutionarily from species C, but it also relates to abundance. So if there's a whole lot of trees of this distantly related species, there might be enough types of pests on them that some of them could spill over to C, but it's much more likely for more abundant, closely related species to do so. And experiments have confirmed that higher host phylogenetic dissimilarity is associated with lower invasion success in this same way. Biodiversity putatively governs a wide range of ecosystem functions, and this includes the ability to potentially buffer against human and animal disease. And this has been discussed in terms of opposing dilution and amplification effects of biodiversity in the literature. And dilution is the first of them, and it is a negative relationship between diversity and infestation or infection success. And it occurs when host diversity decreases the relative frequency of preferred hosts. And so this is something that's been discussed a lot in the Lyme disease literature. So Lyme disease is carried by ticks, and the preferred host of the ticks is the deer. And so sites with a lot of deer will have a lot of Lyme disease because the ticks have a lot of diseased individuals in places that have deer. But when places also have species like mice and opossums, some of those ticks go on to infest them instead of the deer, and the disease actually does a lot worse on the ticks in those vectors, and even more so because opossums eat some of the ticks, so that decreases population levels. So here we're going to see a negative pattern between biodiversity, in terms of 3 species versus 1, and infection success for Lyme disease. And so this has led to the discussion that biodiversity is really great at buffering against infection and infestation in the case of plant pathogens, and promoting this buffering role within the literature. But at the same time, we can sometimes see the opposite. So we sometimes see a positive influence of diversity of hosts on pest success. And this could have happened in the case of Lyme disease if these other species were just as good at hosting Lyme disease infested ticks. Because then the more hosts there were, just like the greater number of potential places where that tick could live and have the disease. But at least in this system, we see more of a negative effect. But we do see evidence of amplification at large scales in something known as the latitudinal biodiversity gradient, which is just a pattern that we see across the world where diversity tends to peak in the tropics and be lower at the higher latitudes. And this is true both for pests and for their hosts. So they're positively related. Where there are more pests, there are more hosts. And here I'm showing that specifically for birds. Where there are more types of birds, there are actually also more types of bird pathogens. So we were wondering how we could figure out when dilution emerges and when amplification emerges, and if biodiversity is the real reason why this is happening. So we thought that the existing literature possessed three main limitations that made it really difficult to tease these things apart and figure out if there's a positive or negative influence of diversity on infection success. So first is a really simple sampling process where it doesn't really matter what the species is that's there, but the more types of species that are there, the more likely you are to get the right one. So this is just a simple sampling process. It doesn't mean that biodiversity has some intrinsic property that prevents infestation or that promotes infestation. It's just fully just the greater your sample size of potential trees, the more likely you will get one that will host a particular pest. Second, we had a density effect, and that is the more types of trees are in a spot, the less easy it is to access maybe the right one. So there could be interference that would prevent the pest from accessing the ideal host if there are a bunch of other types of hosts there. But it's not because they're super diverse that they're interfering, it's just because they're not the focal host. So that would be a mechanism for dilution, whereas this would be a mechanism for amplification. And then finally, a key factor that previous analyses had not looked at is the role of spread. So we know that pests are spreading across space, and the best way to tell where they are going to be in the future is where they are right now. And if you don't control for that in your model, then you might be picking up factors that are correlated with transport, specifically human transport, rather than being correlated with some real property that diversity has on pest success. So you might find that really diverse, dense forests have really low pest loads. And you could say that that's due to their biodiversity, but it could also be because there's no roads going through that forest. So there's no human traffic bringing those pests there. So with my previous work, I was able to tackle all of these things simultaneously and try to separate out that phylogenetic effect. So we hypothesized that when we could look at this phylogenetic diversity separately and model at the same time spread sampling and density effects, we could detect a consistent separable dilution effect, so a negative effect of phylogenetic dissimilarity on pest success. We did this with a model that considered at each site the number of trees, what types of trees they were, and how they were related to the hosts of each pest, the total density of trees, so looking at the potential for interference. And then we're also able to use my spread model predictions from chapter 1 to figure out how many pests did we actually think could get there in the first place anyway, and then model everything else on top of that. And we did this by building two submodels. The first was to just figure out the number of, or the density of suitable hosts at a given site, which we made a function of the number of trees, how related they were to that ideal or focal host community for each pest, and then the total trees to get some estimate of density. And then we then inputted that predicted density from the first model into a model for a given pest species, how likely it was to establish at a site. So it's going to be how many suitable host trees are there, and also how many pests do we actually expect to get there from the spread model. So our best fitting model, incorporating all of these factors, explained 26.5% of the deviance in pest presence across all species. And deviance is a metric that calculates how well you do relative to some constant expectation based on prevalence. So if you know that a pest is present in 70% of sites, a 0% deviance model would just assign a 70% probability to each of those sites. So we get 26.5% better than that. And in this model, spread accounts for the majority of the deviance explained. So you can see these spread terms are really important. This column just shows how much worse we do when we remove that term from the model. And blue are positive terms and red are negative terms. So as expected, places where we expect more spread get more pests. And also as expected, places that have a higher overall density experience more interference, so we see fewer pests. But the really interesting and unfortunately not very important for explaining variation component comes in the susceptibility relationship. So it seems that host diversity has this nonlinear inverted parabola effect. So here I'm showing this line is 0, so this is a positive effect and these are negative effects. So this isn't really dilution or amplification, it's sort of dilution, then amplification, and then dilution again as you get more and more related to a particular focal host. So hosts that are really unrelated to a particular focal host community are bad for establishment, but hosts that are somewhat related to a focal host community actually are the best for establishment. And hosts that are extremely related to the focal host community are again bad for establishment. So this is really novel and interesting. And we're still thinking about why this is happening. So I'm happy to discuss this later if anyone has any ideas. Part of this might be due to generalism. And so if you are a pest that has a lot of different types of hosts, your hosts span a big portion of the phylogenetic tree. And so you're never going to be that unrelated to a given host tree species. So most of your susceptibility relative to other trees is going to be in this region. And if you have a lot of potential hosts, you can probably establish a lot of places and you can probably spread really fast. And this is what we see, that hosts that are pests that have this really general host community tend to spread faster. But this doesn't seem to explain the entire process that's going on here. So there seems to be other factors. And we think this might be related to climatic differences. So the areas that have a lot of these types of hosts might just be really suitable. Like thinking about that latitudinal biodiversity gradient, there might be just certain regions of the US that support a large number of trees types and also a large number of pest types. But this is an open question. So in this analysis, we mechanistically separated the processes involved in establishment and detected a novel nonlinear relationship between phylogenetic divergence and establishment, where amplification occurs at moderate distances, but dilution occurs at really close and really far distances. And at the same time, we see weak density effects and really strong spread effects. And we think this nonlinearity is occurring in part because of this generalism effect, but also potentially due to climatic differences, but we still are not entirely sure. And one major caveat to this approach is that this establishment model comes after we've already fit the spread model. So we could be misattributing some of the variation in establishment that we're seeing as being variation due to spread. We do think, though, that this would, if anything, just decrease the strengths of the relationships that we're finding in this spread model, or in the establishment model, rather than create new relationships that aren't there. So finally, I'm going to be shifting in my last chapter from an ecological prediction focus to a more applied focus, and applying the forecasts of spread from chapter 2 to calculate future estimated damages due to these pest species. And this work was done with Frank Koch and Mark Ambrose. Mark is in the audience today, and I think Frank's going to listen to the recording. And they have done some amazing work, many years of data thinking about these questions for a lot longer than I have, and we've had a lot of great conversations that have been really illuminating for how street trees are really managed on the ground, and just their partnerships with municipalities across the US to create these urban tree inventories have been so incredible. And it's been needed for a long time because a lot of the work is done in forests, but as you'll see, a lot of the damage is happening in cities. So invasive forest pests in the US have already been estimated to cause billions of dollars of damages. And like I said, while most of their hosts are in forests, most of the costs that we're seeing are in cities. And this is because city trees are a lot more valuable. They're also a really high risk to humans if they're dying and could fall on people. And they're also more difficult to salvage. So if a tree, I was gonna say if a tree falls in the forest, if a tree is dead in the forest and it's some land that's supposed to be for timber harvest, it can probably still be salvaged and they can probably still make some money from it. But if a tree is dying on the street, it's not really worthwhile for the municipality at all. They can probably salvage it in some way with some new programs, but it's at a much, there's much less return. So this means that urban trees are causing the most costs, and that these costs are being borne mostly by local governments and residential property owners that have to remove and replace these trees or persistently treat them. And emerald ash borer is the worst of these species, and its rapid uncontrolled spread has led to thousands of trees being removed, not only in the US, but here in Montreal and across Quebec, across Canada. And cost estimates of these damages can help motivate policy. For instance, Brian had been involved in a previous project looking at new legislation to treat wood pallets in the shipping network. And they found that this treatment protocol could reduce or increase avoided damages by up to $12 billion by 2050. So it's really important to show the extent of these damages and how much could be avoided if we take action. So the previous damage estimate for these species comes from Akama and colleagues in 2011, and they used the 3 worst pest species in 3 of the 4 types feeding guilds, so wood borers, sap feeders, and defoliators, to estimate the damages due to all of the pests in the community. And they found that urban trees were the main target of these damages, and so I focus on urban trees here. And due to data limitations at the time, their work was focused only on point estimates of these 3 species, and they also had to assume that trees were distributed in the same way across the entire US. And they also had no ability to produce spatial resolution in their damage estimates. So based on more recent publications and my own work from earlier in my thesis, I was able to improve on all of these dimensions. So this previous work, which I should say Brian was a part of, he did the majority of the modeling component. So they used a Bayesian approach to move from very coarse information about these 3 point estimates to a whole community cost estimate. And all you really need to know about this, the word Bayesian in this case, is that I'm using it because it helps me capture the full uncertainty of the whole distribution here. And the logic is as follows. So they knew that there is some distribution of costs across pests, so different pests cause different costs. They assume most pests do not cause very much damage, but a few are really damaging because that's generally what we see. So there's some negative relationship. They knew that counts can't be negative, so it has to go to 0. They also knew that in general, you can probably assume that pests that are unintentionally introduced in that are forest pests are probably not going to be beneficial. In the worst case, they're going to have no effect. So this all works to restrict the number of curves that could fit this distribution. And then they used information about, they got experts to divide the pests that they knew about into low, intermediate, and this poster pest, worst pest in each feeding guild. And this allowed them to further define the types of curves that could fit this data, because you knew that most of the species were in this category and then less. And so this is a really great way to move from really coarse point estimate information to a whole distribution, and then also to capture that uncertainty through the Bayesian approach. And so I'm going to be using an analogous approach, not for cost, but for mortality of each tree species due to each pest in this updated work. So this chapter combines 4 subcomponent models to produce my cost estimates. First, I have a model for urban trees. Second, I have my spread model for chapter 2. And third, I have a model of mortality of each tree due to each pest, which I just mentioned is the Bayesian approach. And then finally, I have a really simple model for how humans are going to respond to tree death. And the first 3 components of these models are really big advances from the previous work, because we go from assuming all cities have the same distribution of trees to city-specific tree distributions. And we move from point estimates of pest spread for 3 species to all community pest spread from the SDK. And we also get a different type of mortality for each tree and each pest species, rather than assuming constant mortality across space for these pests. So I'll start with the tree model, which I already mentioned is the work of Frank and Mark, who have compiled this really amazing database of urban tree inventories for communities across mostly the northern US, but there's some of the south being represented as well. And in their previous work, they modeled ash, maple, and oak across the eastern US, but I wanted to extend their approach to look at all types of trees across the entire US. So I'm going to be talking about tree genera, or the singular is genus, which is just one level above species for the non-biologists in the audience. So thinking about maples versus Norway maple or something like that. And I broke the trees into small, medium, and large size classes for these models. I focus on street trees because we had the most consistent data for them, and they're likely to be managed the most consistently because they're going to be high priority to remove and replace. And they're almost always the local government's responsibility, so it's easy to assign who pays the costs associated with the damage. we divided the trees into small, medium, and large, and looked first at the total number of trees we predicted in each community, and then built genus-specific models for the number of trees of each genus, which we then used to extrapolate to all 30,000 census-designated places in the US. So we built the models similarly to the GDK, where we were looking broadly at 3 types of predictors. So we had the predicted total number of trees, we had human demographic variables, things like that might moderate human preferences, might vary regionally in terms of what trees people like on the street, and also climatic variables. And actually, similar to the SDK versus GDK comparison, we tested whether we could fit better with a general model, fit across all genera, or if we needed genus-specific predictors. And this is, again, addressing the same sort of question of, are there generalities that help us borrow predictive power across genera, or is it better to really fit to those custom species-specific relationships if they are really idiosyncratic? So here are the fits. Again, I'm showing some R-squareds for small, medium, and large trees. Our predictions are on the x-axis, observations on the y. We do fairly well, and the genus combinations that we don't do very well for are actually really rare species anyway, so there aren't going to be that many trees that we need to predict, and so they're not going to influence our cost estimates very much anyway. And keep in mind that this is an improvement by a lot, because previously we were assuming that trees were distributed exactly how they are in Chicago and Indianapolis alone. And then for the second component, we combine this with our spread estimates, which we forecast first to 2050, so we get our cost estimates up to 2050 from the SDK. And so this allows us to get host distributions and then pest spread, which leads to host exposure to each pest in each community as a prediction. What we then need is how many of those exposed hosts are going to die due to each pest, which we get from another recent publication from Kevin Potter and colleagues, who built this big database of the top pest threats to each tree species, which they grouped into 6 really broad categories here. Some of these are really broad, particularly 25 to 95% mortality. And so we wanted to get an estimate of what the likely mortality is within each of these broad categories, which we added 2 from the original Occam et al. Economic analysis because some species were missing. We assumed these missing species had lower damages than all the ones that were reported as being top threats. Then we broke them into low and intermediate. So if we were just being naive here, we would take maybe the midpoint of each of these bins and assume that that was the severity of each of those pests to those hosts in that severity category. But based on what I was talking about at the beginning of the chapter, there's probably a curve that could describe the frequency of pest host combinations in each of these bins. And it's probably the case that most pests are not going to be that damaging, but a few are going to be highly damaging. So this is why we used a Bayesian approach to look at what that curve could look like across all of our pest host combinations. So here I'm just showing the frequency distribution of pests in all these categories. And as expected, most of them are in that lowest category, but a few pests are really, really damaging. So we ended up using a program called STAN to estimate the curve that could describe this relationship, which is just a program and language for Bayesian computation. And this allows us to get a mean estimate of this curve, as well as a full range of credible intervals. So that's what I'm showing here is our best estimate of what that curve looks like, and then 95% credible intervals so that we can test sensitivity to that full potential distribution of curves underlying this relationship. And so this, again, allows us to move from, so we have host exposure due to each pest and what that's predicted to be to 2050 in each city, and then we know based on that exposure what that mortality is likely to be. Because preventative behavior is much harder to estimate, we then estimated what we believe was the minimal management response required to tree death. And so this is that people are managing only dead trees and they're managing them by cutting them and replacing them. And we got these cost estimates for removal and replacement from the previous economic analysis, but they are really just ballpark figures and they vary a lot. Based on discussions with Frank and Margaret, a lot of this is actually based on how much you need to water the tree. So the southern US, it costs actually a lot more. And again, this doesn't account for any additional preventative measures being taken before the trees die, like cutting non-dead trees or any preventative spraying. We can explore uncertainty in many of the modeling phases within this framework, but the main one that I'm going to focus on is mortality debt because it has the biggest impact on cost patterns. And mortality debt is a term that we created to describe the time period between a pest initially entering a city. And if we know that a pest, for instance, kills 90% of the trees that it's on average from the Potter et al. Inventory, then we want to know how much time does it take between it entering a city and it killing 90% of the trees there. Say 10% of the trees are resistant and they're not going to die. So estimates in the literature span from 10 to 100 years. So we tested this range, and then we just made the simple assumption that that mortality would increase by some constant fraction over time up to the 90% or whatever that maximum mortality would be. So before moving to full cost estimates, it's worth noting how substantial my tree loss predictions are. So I'm predicting up to 1/5 of street trees dying by 2050. And these are mostly ash trees, and they're mostly dying because of emerald ash borer. Here I'm presenting the cost estimates over time for 3 mortality debt scenarios. Oh, and I should mention we call it a debt because it's almost like, for instance, if you have 50-year mortality debt, you're still paying a debt for the species that invaded 50 years ago today, because they haven't fully killed everything they were going to kill when they initially invaded. So mortality debt is a key factor in determining the cost because it ranges by an order of magnitude across scenarios where the longer mortality debts lead to higher costs. And also the shorter mortality debt scenario tracks more closely the short-term changes in spread patterns. So actually in this scenario, emerald ash borer kind of slows down and then speeds back up in this range. So that's why we see this dip. Looking spatially, we see that future damages are going to be primarily born in the Northeast, where we see everything all the time. And that's mostly because emerald ash borer is going to spread there and it's going to kill a lot of ash. And this cost estimate is arguably a lower bound, at least for street trees, because in cities like Montreal, they're doing a lot of preventative management in addition to this cutting. And so that's going to be a key additional factor to consider. Also, because mortality debt has such a huge role, it's really going to be important to figure out what that curve looks like, how long it takes, and how it varies across species. And then finally, I'm only looking at street trees here, but they actually only make up about 2% of all trees in a city. So we really need to know what's happening with those non-street trees. And if we assume that they're managed exactly like street trees are, we could see 1 to 2 order of magnitude higher damages due to these species. So over the course of this degree, I've been examining generalities that emerge at large scales across species invasions, from the ecological to the applied. And these analyses have provided support for the theory that humans are allowing for invasive species distributions and their associated damages become more predictable across contexts. So basically, I've demonstrated support for this anthropogenic replacement hypothesis, where humans are making systems easier to predict at large scales, and use this to my advantage to create higher resolution forecasts, both for establishment spread and future damage. And across chapters, I've used general models that borrow power across contexts to help overcome limited data, whether this was with the GDK, the SDK, my establishment model, or multiple components of my economic analysis. And along the way, I've uncovered a more nuanced role of host biodiversity and improved on existing estimates of countrywide damage due to these species. In the future, I hope to apply these approaches to other systems, and I'm just going to show them really quick right now because I've kind of gone over time. But first, I want to move from a descriptive to a prescriptive angle and look at, still keeping in the theme with large-scale generalities, looking at if there are large-scale generalities in the best management strategies that we can do against these invasive species to limit these costs. And both looking at whether they're generalities and where we apply management, and also how much money we spend on collecting more data and improving our resolution of where pests currently are versus actively treating them. And then finally, I also really like to apply this Bayesian extrapolation method to a bunch of other systems. And it's really cool how flexible this approach is. For instance, right now I'm working with an Australian collaborator to look at predicting population estimates for feral pigs in Australia, because he's really interested in figuring out the risk for future African swine fever in that area. Okay, and with that, it's a wrap. And I have so many people to thank for helping me navigate the roller coaster of these past 5 years, or even like these past 2 months. I've listed a small sampling of you all here, but if you're not listed, it's just because there's so many people, and I'm so grateful. And you all know who you are, and I love you all. And thank you so much for all listening.
That's an excellent job, Emma.
Not just playing an applause sound.
I was trying to figure out how to do applause, but I can't quite figure it out. But maybe, I'm not sure what the best way is of asking questions. Like the hand, I'm not sure if you can see if people raise their hands or not, or
Yeah,
what.
I can bring up the participant list, and also like if people want to bring up their video, you can turn it back on, I think. And then I can unmute people
Maybe you can
as
take everyone off mute first,
okay.
and then they
Okay.
can just ask questions. Maybe
Okay.
that's the
Okay.
easiest.
I'm gonna unmute everybody.
Matt first, the parent, and Piper, and then Jill.
What does he hear from Piper?
I don't know if unmuting everyone was the right strategy because there's I'm gonna mute everybody again, but you can unmute yourself. Sandy has a question, and so I'm gonna unmute him first. Okay, Sandy, you should be unmuted.
Okay. Hey, can you hear me, Emma?
Yeah.
Hey, first I have to say wow, it's so impressive. Your work is just amazing.
Thank
It
you.
really is. And I know so many people in sort of the invasion biology field who have seen your papers that you've published already and so impressed. So, and now seeing more of it, I think your work is really fantastic, just really incredible. And it's been great working with you. So let's see, I have a couple of quick questions. So one, of course, I'm not as familiar with chapters 4 and 5. And my question about chapter 4, maybe I'm a little bit fuzzy still about how you actually did this model fitting. Was it sort of an iterative approach where you basically had these different terms and in different forms? Like I noticed you had this quadratic and linear forms, and then you basically just did like an iterative sort of maximum likelihood where you tried a bunch of different processes in the models. Is that what you did?
Okay, I took out all the math on the lab suggestion, but I can bring it back up now. It's basically it's a big logistic regression that I fit with maximum likelihood, you're right. And I fit it with like a type 3 approach. So I fit the full model and then I tested how different the likelihood was when I removed each term individually. So I can go through it really quickly right now. So this is the first part of the model for the suitable host trees at a site. And it's basically like the number of suitable host trees is, or the probability that a site will have at least one suitable host tree is 1 minus the probability that none of the host trees are suitable there. So that's what this is. And it's a function of, for each type of tree, how many there are, and then a linear and a quadratic version of that susceptibility, which is calculated from phylogenetic distance, and then divided by the total number of trees there. So that's that density effect.
And then how did spread get into that?
So then from there, there's the second component where it's for each propagule that gets to that site, what's the chance that it will establish there? And so the previous model provides this probability of colonization of a single propagule. And then again, we have 1 minus the chance that none of the propagules actually establish. And so the spread model is this PP term up here. So the propagule pressure predicted by my spread model comes in up here, and it's used as a power term. So it's like a coin flipping kind of thing.
And so is all of this stuff basically getting fit sort of simultaneously? Is that the way it works?
Yeah, but the spread model itself is not being fit simultaneously. That's the main limitation. So we have the propagule pressure is just fixed, but all of these terms are fit simultaneously. Yeah, so we fit it with ours Optim package.
Okay, so but the spread part, that's sort of hardwired in there because the species would have to establish first, right? In the initial establishment, right? So are you assuming that each species starts at a certain point in time and then it's spread out, it spreads over time from there?
So we have so from the GDK, we have a relative propagule pressure in each cell, which spans from 0 to 1. So it becomes higher and higher with the more predicted population in that site. So it's non-zero for everywhere where the pest has established in the GDK, but it has different numbers depending on the relative support for the pest being in that site from the GDK. But it is hardwired in, but it's I guess it's proportional to that 0-1 establishment, not establishment, but it has a continuous value from the propagule pressure component of the spread model. but it
Okay.
doesn't change over time. But
Yeah.
yes, yeah, everywhere where we're predicted they're establishing, it's at least some value.
Okay. I mean, to be, it is really interesting. I mean, because, you know, I've racked my own head about this, because I think you're exactly right in your approach in that, you know, when you're looking at sort of the species richness of number of pests per county, there really are kind of two processes that drive that. One is the initial probability of initial establishment, but then once it establishes, there's also then the probability that it's going to spread for any individual county. It's the probability that pest is there is probably affected both by the probability of an initial colonization and then the probability that it would spread there, which is affected not only by the conditions in that county, but also other, you know, nearby areas. So, and from what it sounds like you kind of captured that, but maybe I can look at the details sometime later.
Yeah, I mean, I'll certainly send it to you.
Yeah,
But
it sounds
we
great.
did the best we could, but ideally you would want to do all of that at the same
Yeah.
time.
Oh, and one other thing I was going to say about the susceptibility, which is really interesting, that quadratic relationship. And one thing you might want to take a look at this paper. I was one of many co-authors on a recent paper, Mech et al. It came out Ecology and Evolution, I think 2019, and we were looking not at you know, invasion or establishment, but the impacts of comparing the impacts of various species.
Yeah.
And one of the things we were looking at was the phylogenetics of the hosts in North. We were looking at North American species and the phylogenetic relationship between hosts in North America with the closest host tree in their native region. And it was an interesting thing that we found almost kind of a similar kind of thing. And again, this is different because we're really quantifying impacts, but it might be a similar thing. That is that there's sort of an intermediate sweet spot that if if the host species in North America is really distantly related from it, then the impacts tend to be low. And if the thing that we kind of hypothesized, and others have written about similar stuff, is that if the host is too closely related, then it probably has evolved defenses against similar types of insects. But if it's more distantly related, then not be a suitable host. So anyway, there might be a connection there. Just going
Yeah,
to suggest that.
that's true. And I was looking at that paper, but I didn't look at it closely enough to see that you found that intermediate sweet spot. So that I'm
Yeah.
definitely going to look back at that.
Yeah.
But I was
Oh,
thinking also about natural enemies maybe being something there. And
yeah.
also maybe like sites that only have this precise focal host of a pest could be like really specific climatic, like, I don't know, they're less generally suitable for species in general, and there might be stranger climatically, but I didn't really know how to
Oh,
quantify
and
that.
one last thing, if you don't mind if I can blabber more, but on your chapter 5, which is also really cool, two papers I was going to mention. I don't know if you've seen, there's a paper that Becky Apanchin, Neil, and I published, I think it was 2015. We're looking at all at well, technically we're looking at urban impacts, but we didn't have the really cool host tree database that key thing that way. But we did a similar thing in this 3 or 4 species, the high impact pests, of modeling spread and then discount, spread as they moved into the host range, and then discounting those impacts back to the time of establishment. So there's maybe a similarity there. And then the other thing I was going to mention was in terms of the impacts of the species on tree mortality, that I think you mentioned that you took this information from Kevin Potter's paper, which is really a nice paper, but it's mostly based on sort of educated guesses. And that you might also want to look at, there's this paper that Sung and Fei and Randy Moore and I published using, but again, it wasn't based on urban data, but it's based on FIA data, where we actually quantified tree mortality in relation and the time since invasion. So that might be another thing to look at too. So
yeah, and Brian and I had actually tried to quantify the results of that paper in some way, but the combination of the fact that you're using FIA and we're trying to use the urban inventories where we only have one time point is a little bit difficult, but there might be a way to synthesize that, at least in the Bayesian curve estimation approach, to try to account for both sources of information there. That's a good point.
Yeah, it'd be interesting. I mean, because the interesting thing for some of the, like EAB, it kills pretty fast after lag. I guess what you referred to sort of as this invasion debt. There's this really long lag between the initial invasion and when you, on average, get peak mortality. So interesting.
That's true. But for EAB, we're probably actually closer to this scenario. That's what I'll probably say that in the paper, that like this is probably not the case because the main contributor to cost is going to be EAB, and it probably has maybe a 10-year lag.
Yeah. Anyway, that's really, really cool stuff.
Thanks so much, Sandy.
Yeah.
Anybody else got questions?
I'm pretty sure you'll get many questions later, Emma, probably via individual chats and such. Now, you were mentioning also that, so first of all, again, I think you did an excellent job. And you were mentioning that it would be nice to have a virtual drink with you, even though it is relatively still early in the day.
Yeah. It can be coffee.
It can be coffee. I'm going to have wine because,
Okay.
We're all at home. Well, most of us are probably at home. And so there's probably little, to stop us from doing so. But what I would recommend then is possibly, people can contact you at any point in time and just ask you whatever questions they have. I know I will. But why don't we spend a few minutes and grab a glass of wine or coffee and share it with you virtually.
Sweet. Okay. So I'm going to try to unmute people again. You are all unmuted.
I'll also turn my
nice
camera.
job, Emma.
Thank you.
Good job, Emma.
Mac
Cheers,
has brought me
Emma.
a brownie and some champagne.
Great job, Emma.
Congrats, Emma.
Congrats.
Thanks so
Congrats.
much.
Emma.
That was really interesting.